Hello, welcome everyone out there to today's webinar on the SunScript line of reverse transcriptases from SYGNIS. My name is Armin Schneider. I'm the Senior Vice President of Research at SYGNIS, and it is my great pleasure to introduce to you today Professor Luis Menendez-Arias from the CBM in Madrid and Dr. Raquel Juarez. She developed the enzymes here at SYGNIS. Both of them will give you very interesting talks on features of this new class of enzymes available from SYGNIS now. And as an introduction to Professor Arias, who's really a world-renowned expert on the area of especially the HIV reverse transcriptases, I'll give you a brief biography. He's today research professor at the Spanish National Research Council and the Center for Molecular Biology in Madrid. He started studying biology at the Complutense University in Madrid, received his PhD in 1989, really doing hardcore protein biochemistry, then moved to the US to do his postdoc at the Frederick Cancer Research and Development Center in Maryland. And here really was exposed for the first time to retroviral enzymes and worked 5 years in the US, then came back to Spain and became a staff scientist in 1994 at the Center for Molecular Biology in Madrid, shortly the CBM. That's a very renowned institute here. And he really started digging deeply into researching the HIV reverse transcriptases. In 2001, he became professor and group leader at the CBM and really did a lot of work on drug resistance mechanisms and fidelity of the HIV reverse transcriptases. Before we'll start with the talks, I have a technical remark for our audience. Should you have any questions during the talk or shortly thereafter, you can simply enter this into a little chat field on the lower right-hand side of your screen and hit the send button, and we'll try to answer your questions at the end of the webinar. If not, we'll try to get back to you by email. So with no further ado, Professor Arias, the stage is yours.
Thank you. Thank you, Armin. Hello, everybody. Well, research in our lab has been devoted for many years to study retroviral reverse transcriptases. And today, during this talk, I will describe some of our efforts to generate HIV-1 reverse transcriptase variants with what we believe are interesting properties for biotechnological applications. Let's start by looking a little bit to the essentials of reverse transcription. This is the process that mediates retroviral replication. Retroviruses, as you probably know, are envelope viruses of about 80 to 100 nanometers in diameter that contain within their capsid two single RNA molecules that constitute their genome. These RNAs have a positive polarity and a structure that is similar to that of the cellular messenger RNAs with a cap and a poly A tail. During reverse transcription, the single-stranded RNA is converted to a double-stranded DNA that will integrate in the host cell genome. And this process is catalyzed by reverse transcriptases that need to first synthesize a DNA from using the RNA as a template, and then will go and complete the synthesis by making the synthesis of DNA using the cDNA as a template. Retroviral reverse transcriptases, therefore, have different catalytic activities. They are DNA polymerases, and they have also endonuclease activity. So the DNA polymerase activity, as I mentioned in the previous slide, can use as templates either RNA or DNA, and the endonuclease activity, this is the ribonuclease H activity, allows reverse transcriptase to cleave the RNA when it's part of a complex between RNA and DNA. Besides, reverse transcriptases have strand displacement activity and have the capability of switching templates during reverse transcription. Reverse transcriptases in general come in different shapes and forms. Here I'm showing you the three major structure types that we find among retroviruses. At the bottom you can see the murine leukemia virus reverse transcriptase, which is essentially a monomeric enzyme of 80 kilodalton, which contains both the polymerase and the RNaseH domains. The HIV-1 reverse transcriptase is made of two subunits, P66 and P51, which, P66 and P51 that contain, in the case of P66, the polymerase and the RNaseH domain, while the P51 lacks the RNaseH domain. And then the avian myeloblastosis virus reverse transcriptase has this larger structure with a beta subunit that contains not only the polymerase and the RNaseH, but also the integrase domain. We know quite a lot about the structure of HIV-1 reverse transcriptases because of the interest as a target of antiretroviral drugs. This is the structure of a ternary complex of HIV-1 reverse transcriptase with a template primer. The template primer is shown here with the template in red and the primer in white. The incoming DNTP, which is located in the so-called palm subdomain, is shown in yellow spheres on the top. And then the structure, as you can see, is made of two subunits, P66, which is shown in blue, and P51, which is shown in green. Both of them share the same primary structure, but P51 lacks the RNaseH domain, which is present only in P66. P66 would be the catalytic subunit, which it makes the nucleic acid binding cleft where the template primer binds. Of course, we know quite a lot about reverse transcriptases because of the development of antiretroviral drugs. Since 1987, when the first antiretroviral drug was approved, that was ACT, which was a nucleoside analog, there are a number of different combinations, different drugs that have been developed, and for many years, the treatment with those drugs would develop resistance, would generate resistance, and variants of the reverse transcriptases would contain those mutations that confer different specificities to those enzymes. Today, eventually, antiretroviral treatment is based essentially on the combination of three nucleoside and non-nucleoside, three reverse transcriptase inhibitors, which are tenofovir, efavirenz, and FTC, or emtricitabine, and this is the most widely and popular treatment used today. So even today, the reverse transcriptase is a major target of antiretroviral intervention. For many years, we have been interested in understanding the mechanism of drug resistance in HIV-1 reverse transcriptase, but also how reverse transcriptase controls the specificity of nucleotide incorporation. And back in the early 90s, we were working with different enzymes, and eventually we found that tyrosine 115, which is an amino acid located below the incoming nucleotide, below the ribose ring of the incoming nucleotide, which is shown in yellow in the figure, this amino acid was important for the discrimination between RNTPs and DNTPs. And in fact, if the aromatic side chain is removed and substituted by, let's say, valine, alanine, or glycine, the enzyme loses its ability to discriminate between this type of residues. In addition, we also found then that substituting tyrosine by other residues will decrease the fidelity of the reverse transcriptase. But also, this decrease of fidelity is usually accompanied by a loss of activity. So sometimes fidelity and activity are sometimes linked and are difficult to keep as separate entities. These are general studies that were performed essentially with reverse transcriptase variants, which derive from the initial strains that colonized or infected in the Western world. And those strains are classified generally as HIV group M subtype B strains. And those are usually some of the strains that were initially characterized, like BH10 that you can see in the bottom of this phylogenetic tree. Those strains are the ones that are used as a reference as prototypic strains for HIV-1 reverse transcriptase and for all the phenotypic and biochemical studies that have been described in the literature. However, these are not the only variants that you find in nature, and there are other other viruses that contain different RTs which colonize different areas of the planet, and in some cases become apparent or can cause also disease. One of them is the HIV-1 group O. The group O is a clade that infects mainly people in Central Africa. The first cases in Europe identified were people coming from Cameroon. And back in 1997, we had the opportunity to study one of these variants and to isolate and obtain the reverse transcriptase of this HIV-1 group ORT. In general, HIV-1 group O reverse transcriptases differ in about 21% of the amino acid sequence comparison with the typical or prototypic BH10 RT. But an interesting clinical feature of these enzymes is that the group ORT is naturally resistant to non-nucleoside RT inhibitors. Well, back then we were interested in those characteristics, of course, but back then we realized also that the wild-type HIV-1 group ORT that we isolated from one of the Spanish patients that were eventually people coming from who got infected in Equatorial Guinea, a former Spanish colony, that belonged to the so-called S strain, that we found that this variant RT was a little bit more resistant to urea treatment than the prototypic BH10 RT. So that put us on the clue that we could perhaps think about this enzyme as a more stable RT. And in fact, years later, because eventually this RT was difficult to purify in relatively high quantities, we could demonstrate that the group ORT showed relatively high thermal stability, or at least high efficiency at high temperatures. In this figure, I'm showing you an example of in which you can see that at 57 degrees, the amplification of tubulin cDNA of this fragment of about 1200 base pairs, this is not possible to observe such an amplification with the mutant leukemia virus reverse transcriptase, but it is possible to obtain this amplification with the two HIV-1 RTs. Interestingly, at temperatures above 66 degrees, the HIV-1 group ORT showed higher stability, and therefore this was a good indication of its good properties in terms of efficiency at high temperatures. However, it has been widely recognized that HIV-1 RTs are less faithful in general than the typical oncoretroviral RTs. The old literature states that they are about 10 to 15 times less faithful. So we went into further analysis trying to improve the fidelity properties of the HIV-1 group ORT. And for this purpose, we use a lot of information that that different labs have produced, including ours, about the effects of single amino acid substitutions in the fidelity of the prototypic HIV-1 group M subtype B RTs. And this is a summary of some of those studies in which what you can observe is that the big bars that represent enzymes which are mutants that have high accuracy of DNA synthesis. High accuracy not always relates to higher efficiency, so both factors have to be balanced. So some of those mutations produce enzymes that have high fidelity, but however they have low specific activity. Keeping in mind both ideas, we selected 3 mutations that we thought would be good candidates to improve fidelity without altering activity. And these mutations were K65R, V75I, and arginine R78 alanine. In the case of lysine 65, this residue is located at the DNTP binding site, and in fact, it interacts with the phosphate group of the incoming DNTP. That's what you can see in the panel B. However, the other two residues, valine 75 and arginine 78, contact the template primer, the template position +1, so the complementary position where the DNTP binds. Well, we made those mutants that contain those mutations in the context of the HIV-1 group ORT, and we check their thermal stability, in this case using a different type of assay. In this type of assays, what we did was to incubate the enzyme for 5 minutes at the indicated temperatures and then measure residual activity at 37 degrees. The major conclusion of this analysis was that at least the 4 enzymes that appear indicated with the yellow arrows, they keep significant thermal stability. However, and significantly higher stability than the BH10 wild type RT that you can see, for example, the differences at 54 degrees. And in the fact, R78A and the double mutant B75I R78A showed very poor stability. I should say that in this case, R78A was a deleterious mutation for activity. So again, not always fidelity and activity correlate well. These data were corroborated by looking at the effect of temperature in the efficiency of cDNA synthesis in RT-PCR reactions. Again, we observe good efficiency, for example, in the amplification of actin at 68 degrees with the 3 enzymes marked with the green ellipse. And here we have K65R and K65R B75I as 2 mutants that show increased or good thermal stability in these assays. But again, we come to the major issue: how we measure fidelity, and the key. And it's important for us to have an assessment of the fidelity of those enzymes. And fidelity can be measured in using different types of assays. We can use enzymological assays in which we measure catalytic constants for the incorporation of nucleotides. We can do primer extension assays in the absence of one nucleotide. But one of the best assays for assessing fidelity in our hands is this forward mutation assay, which was initially designed by Bevenek and Kunkel years ago. And in this assay, what we do is we use a gap DNA substrate, which is made of M13 double-stranded DNA, in which we delete one of the strands of the LAC-C gene, and this strand is filled with the RT to complete the synthesis of this double-stranded DNA that we then transform in E. coli, and after transformation, we can plug the results of the transformants on XGAL and IPTG to obtain blue or pale blue or colorless plaques. If the RT during the process of filling up the gap makes a mistake or introduces a mutation that causes damage in the phenotype of LAC-C, we will see plaques which will be pale blue or colorless, which can be selected, counted, and eventually we can obtain the DNA and sequence identify the mutation that caused this defect. Using this type of assays, which are time consuming anyway, we can assess the fidelity of many mutants, that we have done so with a number of mutants. And just as a summary, here I'm showing you 2 good examples of the effects of fidelity of those of some mutations that we introduced in the context of the HIV-1 group ORT. We should note that the differences between the HIV-1 group ORT, the wild type, and the MULB in terms of fidelity are not as huge as it was initially assumed. It was assumed to be 10 to 15 times, tends to be more like 5-6 times. But anyhow, if we introduce mutations such as K65R or the double mutant, K65R B75I, we can obtain RTs with R is even more faithful than the wild type MULB RT. And on the other hand, if we introduce mutations that eliminate the RNaseH activity, like D443N or E478Q, the one shown in blue in the slide, we obtain RTs with similar fidelity compared to the wild type MULB. So far so good, but we were interested in improved even further the thermal stability of the HIV-1 group ORT, or at least understand the basis of this thermal stability. For this purpose, we made a three-dimensional structure, a molecular model of the HIV-1 group ORT, shown below, based on the structure of the wild-type subtype B HIV-1 reverse transcriptase. And using this model, we measure the contribution of both subunits to the stability, the interaction between P66 and P51. This is shown in panel A, and that shows no differences between both. So the interaction between both subunits is not critically different between both enzymes. However, if we look at the interactions between the RT and the template primer, there are large, significant differences. So the group O makes stronger interactions than the typical subtype B RT. So the basis for the higher thermal stability of the group O RT is located or refers to this higher ability to interact with the template primer. But we can still improve this interaction by looking at how these enzymes interact with their corresponding nucleic acids. And we look again to the model and the structure. And what we can see here, this grid shows in red the positions where the interaction between the group ORT and the template primer is stronger, and in blue, the positions where the interaction in the subtype B with the DNA is stronger. And there are differences. And interestingly, when we look at the subtype B, the differences are consistent with differences in the sequence of the reverse transcriptase. While residues 355 to 361 in subtype B contain this sequence AR, MRGAH, in group O, the sequence is completely different. So what we thought is maybe we could still improve the group ORT by changing those residues, putting those residues that are present in the subtype B enzyme into the group ORT. And for this purpose, we generated a series of mutants that include this O3M-RT, which contains the three central residues in this structure. As you can see in these RT-PCR reactions, you're looking at the effect of the temperatures, we observed that the 3M enzyme was able to amplify at 78 degrees and was the best one, performed better than the wild-type group ORT. And we went even further and introduced, in this context, all the mutations that we had some hint that could produce an increase in fidelity. However, in many cases, the increase in fidelity that we expected was not really accompanied by an increase or maintaining the thermal stability. And unfortunately, some of the mutations that were supposed to increase the fidelity were not so good in terms of maintaining the thermal stability. Only a couple of cases, the 3M and E478Q, which is the RNaseH minus variant of this 3M enzyme, and the one that contains the insertion, they retain similar activity as compared to the basic 3M enzyme. When we look at qPCR assays using these enzymes, again, results are similar and consistent. The best enzymes in these qPCR assays were those that we designate as 3M, and the 3M with the RNase minus mutation, the E478Q. And as we expected, as we hypothesized, these mutations, these two enzymes were the ones that showed the best or the highest affinity for the template primer. And those two enzymes were really the best ones in terms of further development or in terms of increased efficiency at high temperature. When we look at fidelity of those mutations, eventually the fidelity was rather similar to that of the wild type, a little bit higher in the case of the RNaseH deficient mutant, and a little bit lower than the MULV-RT. However, we should note here that these assays are carried out at 37 degrees, which is not the working temperature that we tend to use with the thermostable enzymes. Interestingly, although we know that the HIV-1 group ORT shows significantly higher thermal stability than the phenotypic HIV-1 group M subtype BRT and slightly increased fidelity of DNA synthesis in DNA-dependent reactions, we can observe that in fact at 55 degrees we can increase with the wild-type RT 5 times the fidelity of the enzyme in the LAC-C assays as compared with the 37 degrees for the wild-type RT. So just to summarize the last part, I would say that large increases in thermal efficiency at high temperature can be achieved by improving interactions with the template primer, for example, at the connection subdomain, that amino acid substitutions such as K65R, K65R, B75I, and the RNaseH inactivating mutations produce 3 to 10-fold increases in fidelity. However, sequence context is important, and those mutations seem to destabilize the RT when the connection subdomain mutations are present. So I think this is all, and perhaps we should go to the next part.
Hello to everybody. I'm Raquel Juarez, the project manager at SYGNIS, responsible for the SunScript product line. I'm going to continue with this part of the webinar. This product line consists of 3 different products at this moment: the enzyme itself, SunScript Reverse Transcriptase, in 2 different versions, RNaseH Plus and RNaseH-, and a first kit based on this enzyme, SunScript One Step RT-PCR kit. Our enzyme, SunScript Reverse Transcriptase, is a novel, highly thermostable reverse transcriptase engineered from the well-characterized HIV-1 group O RT. It allows reaction temperatures up to 85 degrees and offers improved efficiency of cDNA synthesis. As I have introduced, it comes in 2 different versions, RNaseH Plus and RNaseH-. The competitive advantages and benefits of this new enzyme are: it is a reverse transcriptase with strong displacement capacities. It is highly thermostable, active at 25 degrees to resolve even the most complex RNase structure. The optimal reaction temperature for this enzyme is 60–65 degrees. High reaction speed of more than 30 bases per second. It produces high yields of first-strand cDNA. It produces complete reverse transcription of very long RNA molecules. It possesses high sensitivity down to picograms of starting total RNA, and the obtained yields are highly consistent, giving reproducible results. Regarding the 2 different versions, RNaseH Plus has RNA and DNA-dependent polymerase activities, as well as RNaseH activity. It is described that RNaseH activity is beneficial for the template switching activity, so this version is more adequate for those users looking for this application. And the minus version lacks the RNaseH activity and is more adequate for some applications requiring high yields of full-length cDNA synthesis or long RNA molecules with complex secondary structures. It is also recommended for applications requiring greater gene representation. The basics of the reverse transcription reaction are depicted in this scheme. In the first step, the oligos are hybridized to the RNA strand. They can be oligo-DT, gene-specific primers, or random primers. In a second step, the reverse transcriptase binds and synthesizes the complementary cDNA strand. And finally, the reverse transcriptase is inactivated and a hybrid molecule, RNA-cDNA, has been obtained for subsequent analysis. And now I'm going to show you some figures that pictures the main special features of SunScript. In this one, we can see how both versions of SunScript allows reverse transcription up to temperatures of 85 degrees. After the reverse transcription, a PCR has been performed for a fragment of 305 base pair for GAPDH. Although the recommended temperature for most reactions is 65 degrees, higher temperatures are possible, obtaining good final yields. The higher reaction temperatures can be positive for transcription of RNAs with complicated secondary structures. In this slide, we can see the high sensitivity of both versions of SunScript over a wide range of RNA concentrations. This reaction was performed at 70 degrees, and again the subsequent PCR amplified a small fragment of the APDH gene. We can see how the reaction is effective from total RNA amounts from 1 microgram to 100 picograms. This figure is an example of detection of long reads. 3 replicates of each version of SunScript have been performed for each example. The one at the right side is the detection of a small fragment of the gene DNA-dependent, which is located at a distance of 8.5 kilobases from the poly-A tail. The amplification of the fragment confirms that the reverse transcription has been effective at least up to that size. On the left side of the slide, we can see other similar examples with the gene of nebulae. In this case, the amplified fragment is located at a distance of 16 kilobases from the poly-A tail of the mRNA. For this experiment, the RT reaction time was 60 minutes. In this slide, we have an example of the efficiency and speed of the RT reaction. Here, both versions of SunScript has been tested for the amplification of a small fragment located at 11 kilobases from the poly-A tail of HAART mRNA. Several reaction times for the RT step has been tested from 60 minutes to 5 minutes, showing no reduction in yield or efficiency for the shortest reaction time of 5 minutes. Regarding the fidelity, we have performed a comparative experiment with other commercial competitors in a MySeq platform to determine comparative error rates. The experiment is based on the reverse transcription of beta-actin with different thermostable RTs at 65 degrees and PCR amplification. The sequencing was performed with Illumina MySeq platform. The result reflects the combined error rate of the RT enzyme, the PCR step performed with a high-fidelity polymerase, and the sequencing machine MySeq, which gives 0.4% nucleotide error rate. The results of this experiment gave the following conclusions. Both versions of SunScript have similar error rates. SunScript has the same error rate than competitor T and a clearly lower error rate than competitor M by almost 0.1%. Despite its origin as a HIV RT, SunScript has good fidelity at high temperatures. Both versions of SunScript Reverse Transcriptase are commercialized into different formats. We are currently commercializing into different formats, but we are changing to the ones that you have here in the slide from next Monday. A 10 reaction format with a price of 60 euros, a 50 reaction format with a price of 160 euros, and a 200 reaction format with a price of 530 euros. Prices don't include shipment costs. You can order this format through our online shop at SYGNIS website. And now I'm going to present you our most recent product of the SunScript line, which has just been launched in the market last September. SunScript One Step RT-PCR kit. The key advantages of this new kit are: it is a fast, specific and sensitive system for endpoint RT-PCR reactions. It performs both the reverse transcription and PCR in the same single tube. It allows one-step reaction to minimize contamination. It is a convenient system to process multiple samples. Given that it is based on SunScript, it is an excellent choice to amplify difficult RNAs. It produces high reproducibility of reactions, and it is robust and reliable. In this slide, you have a scheme on how the one-step reaction performs. In a first step, the specific oligos are hybridized to the RNA strand, and then the reverse transcriptase synthesizes the complementary cDNA strand. After that, the reverse transcriptase is inactivated and the tag polymerase is activated by heating the reaction at 95 degrees. Then the tag polymerase amplifies the desired fragment of cDNA by PCR using the same oligo of step 1, the reverse, and also the corresponding forward oligo. And now I'm going to show you some figures that picture the special features of this new kit. Based on SunScript Reverse Transcriptase, the SunScript One Step RT-PCR kit is able to perform the RT reaction up to temperatures of 85 degrees. Although the recommended reaction temperature for the RT step is 60 degrees, it can be also adjusted to higher temperatures up to 85 degrees if needed, especially for detecting difficult RNAs with high degree of secondary structure or high GC content. In this example, specific primers for detecting a fragment of the GAPDH gene have been used. In this slide, we can see that SunScript One Step RT-PCR kit is able to detect genes with different expression levels. Specific oligos for different genes have been used: GAPDH, ERG, GU, SV, CLTC, and actin. And you can see how the kit gives good yields and detection levels for any of the tested genes. Here you can see that SunScript One Step RT-PCR kit has good and enough sensitivity to detect amounts from micrograms to picograms of starting total RNA. In this example, specific primers for GAPDH gene have been used. A key feature of this kit is its ability to produce highly reproducible results among different samples or replicates. Here you are an example on how the yields and detection looks for 4 replicates of different genes: GAPDH, actin, and CLTC. And you can see how reproducible the result is among replicates, showing similar sensitivity and yield, which makes this kit the best choice for multiple sample processing with minimum deviations. And finally, here you are an example on how the SunScript One Step RT-PCR kit is able to effectively amplify difficult RNA molecules. For this experiment, 4 different specific primer pairs were used to amplify different parts of an RNA fragment with a GC content of 75%. Even performing the RT step at 60 degrees, the 4 oligo pair produced specific amplification bands despite the high GC content of the template RNA. The SunScript One Step RT-PCR kit is commercialized in 2 different formats: a 25 reactions format with a price of 161 euros and a 100 reaction format with a price of 541 euros. Prices don't include shipment costs. You can order these products through our online shop at SYGNIS website. Well, I hope this webinar has been useful for you to understand the main features of our enzyme SunScript and products based on it. And now, if you have any questions, please feel free to ask. Thank you.
So, Luis and Raquel, thanks a lot for this very thorough overview of both the scientific and applied aspects of this novel engineered class of HIV-RTs. I might add that these are the only reverse transcriptases on the market available derived from the HIV virus. So far, all other RTs are either MMLV or AMV. Okay, we have some questions here, and maybe I'll read them out and just direct them to the ones I think might best be able to answer them. So for Raquel, is a question: if you have a new unknown template which you have never amplified before, which temperature would you recommend to start with?
Well, we also recommend to start with temperatures of 60-65 degrees, but you can adjust to higher temperatures if needed, from 5 degrees to 5 degrees, up to 85 degrees as a maximum.
Okay, and another one for you, Raquel. Have you compared the SunScript enzyme with superscript for Fisher?
No, we haven't performed a direct comparison, but superscript is an enzyme working up to temperatures of 50-55 degrees as a maximum. So the advantage that SunScript gives is that you can use even higher temperatures with a maximum of 85 degrees, which gives an extra point if you are interested on amplify difficult RNAs with complex secondary structures or high GC content.
Okay, thanks. I have a rather more complex question for you, Luis. That is, does SunScript add a few bases of C at the 3' end of the newly synthesized cDNA, as many other RTs do?
Okay. What we know is that it may add one nucleotide. We have seen many times this in the case of HIV. However, the addition of non-templated nucleotide is really it has very low activity in comparison to the polymerization activity. So it's not something that occurs always. It depends on the conditions, and eventually for HIV-1 is an A more than a C, what is usually incorporated. And I would say that although we didn't test this specifically, we haven't really done extensive analysis on that. This has been reported earlier by other groups that HIV-1 group or HIV-1 reverse transcriptase, the prototypic RT, can do this type of reactions. Although I'd say that only in certain conditions and with lower efficiency than the typical polymerization activity.
Okay, thank you. There's another question for you, Luis, I think, regarding question to the relative template switching activity of the RNaseH plus and minus forms.
Okay, yeah. Again, the reference should be HIV-1 reverse transcriptase in general, and of course it has template switching activity. In principle, even the RNaseH minus or plus doesn't make big difference. So in both cases, you would expect template switching activity, only that in the case of the RNaseH minus, probably will be a little bit less efficient, but eventually you have the template switching activity anyhow. And we should expect that for SunScript enzymes also.
Okay, and that's maybe a question to both of you, whoever likes to answer it, and a more detailed question on the comparison available for fidelity of different RTs at high temperatures.
Okay, there are no reliable data on that in the literature, because any estimate of fidelity has limitations. It's not so easy. As I mentioned in one of the slides, we know that at 50 degrees, the HIV-1 reverse transcriptase and the group O enzyme has about 5-fold increased fidelity in comparison with the wild-type enzyme. And when we did those assays, we compare the fidelity of this enzyme at 50 degrees with the fidelity of one of the typical thermostable RTs that are commercialized, and it was rather similar. So you have to be very careful and perhaps compare different systems before making a clear statement on that.
Okay, thank you. With one last question, I guess, for Raquel on the details of this long transcript experiment. And someone from the audience asked whether you have PCR'd out the entire long transcript to eliminate the concern of random primers.
We haven't performed that kind of testings, no. We have only performed these experiments I have shown of a small fragment located at a certain distance from the poly-A tail, but not the amplification of the entire transcript.
Mm-hmm. But I guess there's no doubt that the PCR comes from the long transcript and proves the transcription of this reverse transcription of this long transcript from the poly-A tail. Okay, so we have no more questions. So again, I would like to thank today's speakers, Luis and Raquel. And I would also like to thank the audience for their interest and their attention and contribution. And I just want to finally highlight that, as we had mentioned, the products are available through our web shop, but as well through many distributors worldwide that are distributing our products. Information on our distributors can also be found on our